All right, everyone, I want to go ahead and get started. Welcome. Thank you for joining us for this webinar, Neuroscience and Cannabis, Implications for Law and Policy. My name is Susanna Baruch. Excuse me. I am the Executive Director of the Petrie-Flom Center for Health Law Policy, Biotechnology, and Bioethics at Harvard Law School. This event today is part of Petrie-Flom's collaboration with the Center for Law, Brain, and Behavior at Massachusetts General Hospital. Many thanks to our colleagues at CLBV for bringing us this event. We are thrilled to have Dr. Yasmin Hurd with us to explore the science of cannabis, CBD, and the future of substance use disorder treatment. In a moment, I will introduce our senior fellow, Stephanie Tabashneck, who will lead and moderate our event. But just first, a few things of housekeeping. Feel free to submit questions. We'll have an extended Q&A at the end of the event. Use the Q&A feature found in the meeting controls at the bottom of your screen. You can join the conversation or even submit questions on Twitter or X at Petrie-Flom. We will share the fully captioned event video with all registrants within a couple of weeks. If you have any technical issues during the event, please email us at petrie-flom@law.harvard.edu. And if you like today's event and you're interested in other Petrie-Flom Center events, please subscribe to our newsletter, look at our website, and read our blog, Bill of Health. So with that, I'm introducing to you one of my favorite people, Dr. Stephanie Tabashneck, a forensic psychologist and attorney who is a senior fellow in Law and Applied Neuroscience at Petrie-Flom, part of the collaboration I mentioned between CLBB and the Petrie-Flom Center. Stephanie is known for her own important work at the intersection of psychology, policy, and family law. She's both a psychologist and an attorney, and that shows in all of the work that she does. Her practice areas include forensic psychological assessment, child custody, and criminal forensic evaluations. And she's a nationally recognized author and presenter on a range of topics. So with that, thank you for joining us, and I turn the mic to Stephanie.
Thank you, Susanna. And if you don't mind just stopping sharing, I'll pull up my slides. And I am here today with Dr. Yasmin Hurd, who is fabulous. She is a neuroscientist and researcher. She's an expert in cannabis. She's very widely published, and she's the director of the Addiction Institute at the School of Medicine at Mount Sinai. You are in for a treat. Usually we have this annual event on 4/20. But 4/20 was Saturday, so instead we're doing 4/18. So I'm going to just speak briefly about the current legal landscape, and then we're going to go sort of deep into the neuroscience of cannabis. So right now, cannabis is scheduled as a Schedule I drug, and that means that there's no currently accepted medical use and a high potential for abuse as designated under the Controlled Substances Act of 1970, which is federal law. And other drugs in this category include things like methamphetamines, heroin, cocaine, and so on. So cannabis continues to be federally prohibited. There's a number of bills that are pending at the federal level that is looking to change this, and in particular, the Cannabis Administration Opportunity Act and the Marijuana Opportunity Reinvestment and Expungement Act. And on the state level, there's been over 1,400 bills that were proposed in state legislative sessions during last year. So there's a ton of movement here. And this was a Dear Colleague letter that was sent by the Senate leader, Senator Schumer, as well as others, just recently as part of a endeavor to pass federal legislation over cannabis and removing or descheduling cannabis. So basically saying the war on drugs has failed. Let's leave this to the states. 90% of American adults believe that cannabis should be legal. 50% of self-identified conservatives believe that there should be either legal. It should be legal for personal or medical use, and also saying that this is generating a lot of revenue that the federal government is missing out on. And also that this is a sort of a safety issue. So right now there's no really not any federal regulations of cannabis. And there's an argument that this should be subject to the same high regulatory standards as other, sort of substances like alcohol and tobacco, and also preventing children and adults who are younger than 21 from using cannabis. So just briefly, we'd be remiss if we didn't talk about the war on drugs and the racism that goes into the prosecution of drug crimes. So we saw a radical exponential increase in people being prosecuted for drug crimes from 1925 to 2016. Cannabis arrests are about 1/10 of arrests nationwide, and the vast majority are for just possession only. We know that at every touch point of the justice system, there is racially disparate enforcement, everything from just who is stopped or detained for a crime or a drug crime and what kind of sentence that they receive. And in every state, Black people are more likely to be arrested for cannabis possession than white people. And this is also in states where they've legalized cannabis. In some states, 90% of drug prisoners are Black, and maybe up to 57 more times than whites to serve prison time for drug crimes, even though we know that drug use is pretty equal across race, including cannabis use. And this is also an issue in child welfare, where parents who are Black and brown are much more likely to be drug tested at birth, and where women who test positive for cannabis at birth may have their children removed, which is more likely to happen if a parent is Black or brown. So I'm going to kick this over to Dr. Hurd, who is going to do a deep dive into the neuroscience of cannabis.
Thanks very much, Stephanie. Really appreciate it. It's great to once again be part of this. I'm going to share my screen. Be part of this 420, which really is for 18 this year, as you said, and I hope that we can have interesting discussions as usual. I always and you've already emphasized the aspects of law and discover a couple of things, but disclosure, I'm not a lawyer, but the issue is, does neuroscience and what we've learned about cannabis, its implications for law and policy that I think are really important. So, one of the things, and Stephanie has gone over a couple of things, but for me, just again, giving perspective on this plant that has been in the US, from perhaps the 1600s, and just what the history had been. Cannabis actually early in our history was used for quote unquote medicinal purposes. When you look at a lot of this, though, a lot of it had alcohol too. But in the 1930s, with a number of things, with more cannabis use, especially from Mexican immigrants at that time that were coming in, there was this marijuana tax act that prohibited marijuana federally, but it still allowed medical use. 1950s came along, and then there was mandatory sentencing, even 2 to 10 years with a fine of $20,000 for the first offense of marijuana possession. And as Stephanie showed, the Controlled Substance Act that's made cannabis a Schedule I drug, just as heroin in 1970s. And the whole issue of the war on drugs that started when Nixon rejected the Schaefer Commission report to remove cannabis as a Schedule I drug, that led to, as she showed, this mass incarceration. But it also in the 70s began with this liberate marijuana, push, and many states did begin to decriminalize marijuana, with California becoming the first state where voters actually approved cannabis for legalization for medicinal purposes. And then many other states followed in terms of legalizing for medicinal or recreational use. In 2018, the Farm Bill that legalized the production of hemp, which is the cannabis plant with less than 0.3% THC. So what does that all mean? It means, as Stephanie pointed out, this complex legal status in our country, where we've gone from such prohibition to now, the majority of states have legalized some form of either recreational or medicinal use. And now, but it's still on the backdrop that cannabis is still an illicit substance of use federally. So very, very complex. But so is the plant complex, and all these sociopolitical changes. It's I think that people don't understand this plant. This is, as I said, a plant very complex with over 140 cannabinoids that we still don't know their activity. We studied for the most part THC and another cannabinoid that's being studied in more recently, cannabidiol CBD. But there are a lot of other chemicals, over 500 chemicals, that might also have biological activity that we still don't know about. And today, the plants that are being consumed for the large part are not the original plants that were there in terms of people talk about old indica and sativa. Most of the plants there have been mutated. They are hybrid strains. But it's also on top of this historic concentrated high potency products that are there. So clearly people are not only smoking joints, they're vaping, they're now edibles, they're sublingual tinctures that perhaps more, and even the dermal patches for more medicinal implications. Also, we have now these highly concentrated, excuse me, products like the shatter, and how they're using them, the dabs, that will even give up to 90% THC. So we're not talking about the cannabis plants that are the THC contents in the original cannabis plants that were about 2 to 4% THC. And I just wanted to emphasize that as the market has grown, the flower products that were there, they're still prominent, but the concentrates are taking over, and the access to this cornucopia of products is very high in our society. As I said, the Farm Bill was an important bill because it did legalize hemp, less than 0.3% THC. But that has also had a lot of loopholes, because people then use the CBD, the hemp-derived CBD, to chemically convert it to THC analogs. And those THC analogs can have potent effects on the brain as the delta-9 THC, even the precursors that they will heat and the chemicals that they use to make this conversion. That's not being done in the plant, it's being done synthetically. And these chemicals can also have toxic effects. They've even now, when we have identified other very rare cannabinoids in the plant, these can now be made synthetically, and they can have potency even greater than 30% of more potent than delta-9 THC. So the complexity of this plant, the history and legal status, is also integrated with the complexity of where do the cannabinoids have their action in the body. So this and our endogenous biology and the mechanism of action. So when we think about the cannabinoid plant, obviously the people who are consuming it for recreational use, it's delta-9 THC. That's associated with the reward, the high. And many people also believe, obviously, that the THC has medicinal properties. THC binds to our endogenous cannabinoid receptors. So this is, as you can see, the schematic on the left, representing cannabinoid receptors in the body. And you can see they're really abundant throughout the body. Practically every organ expresses these cannabinoid receptors. In the brain is really intense, especially for the cannabinoid subtype receptor 1. These receptors are not there for THC, which is a partial agonist of these receptors. It's there for these endogenous cannabinoids, these endocannabinoids as they're called. And they're basically synthesized on demand, and they're synthesized from lipid precursors. But once again, people think, oh, endocannabinoids, they're natural, they're organic, we know so much about them. That's not true. These endocannabinoids belong to this huge, complex lipid system that we still have very little clue about them. We're learning every day. So we know that there are these endogenous endocannabinoids and part of these lipid systems. They're critical for numerous biological processes because, as you can see, the receptors are expressed throughout the body. So energy metabolism, appetite, immune function, neurodevelopmental, as I'll talk about, cognition, pain, our sleep-wake cycle, and especially stress and emotion regulation. So in the brain, as a neuroscientist, I'm biased. That's one of the things, the organ that I studied the most. And the cannabinoid receptor, especially the CB1 receptor, is the most abundant receptor on our cell surface of the surface of our cells in the brain. And these are expressed throughout the brain in so many brain regions relevant to in the cortex for cognition, motor function, memory, emotional regulation. The cannabinoid, the endocannabinoid system in the brain is a enormously important neuromodulatory system. It fine-tunes how cells communicate. I'm not going to go into the minutiae of this, but the receptors and the enzymes that make our endogenous cannabinoids and break them down, they are in, there's a symphony. And like I said, they play a really critical role in this, in modulating synaptic plasticity. So clearly, we have always been at the time, and you can see from the historical perspective that I showed before, that this debate about cannabis has existed forever. On one hand, there's people who talk about that it is of no harm, it's healthy, it's organic, it's not addictive, it alleviates their anxiety, basically it can treat every drug. And as Stephanie indicated, the mass incarceration, especially of Black and brown individuals, this racial bias and the inequities that came with it, also, if you legalize, then you can get rid of that. That was what the hope was. And obviously, she also showed many people, we can make a lot of money on this cannabis industry. But the other side is the psychopathology that might exist with cannabis use, the addiction risk, and that it indeed may promote anxiety and it might impact the developing brain. And many communities are worried that the cannabis industry and the illicit market in particular, especially with these high potency products, can harm their communities and even our healthcare system that's already on the brink. So I'm going to talk about cannabis and or at least the neuroscience also of cannabis in regard to some of these issues. And excuse me, I've been coughing a lot the past few weeks. But I'll start with just the basic of understanding when someone consumes THC or consumes cannabis and THC as the main psychoactive cannabinoid associated with the high. We have this, our natural reward system, neurotransmitter dopamine. When cannabis THC binds these cannabinoid receptors, changes the activity of these dopamine cells, and dopamine levels are increased. Someone may feel euphoric, like most drugs of abuse. And when they're not there, if they've been using a lot, and when the drug is no longer there, they may even start to become dysphoric. And people have done imaging studies in healthy individuals and others and giving THC, and they see that indeed dopamine does increase. And as the dose and concentration of THC increases, so does the dopamine levels. But the acute effects is not, and especially the acute effects of just, infrequent use is not really the issue that has been the main thing, even though obviously, aside from euphoria, can lead to memory impairment, obviously motor impairment in terms of driving, but it's this persistent longer effects that may be a problem in terms of sleep impairment, and even leading to an actual cannabis use disorder itself, where like other substances, it's a clinical diagnosis of cannabis being used in ways that is not desirable for the individual's long-term health. And it's associated into all of these criteria. I'm not going to go through them, but you can have a mild, moderate, or severe cannabis use disorder. And it's estimated that approximately about 10 to 30% of individuals who use cannabis will develop a cannabis use disorder. We also know in looking at the brain, these schematics just to show that there are structural neurobiological alterations that actually look like what happens when in other substances of abuse. And the same thing on the functional level, such as changes in their striatum, this area of the brain, the nucleus accumbens, that's really important for reward. The amygdala, critical for emotional regulation, our prefrontal cortex for working memory, attention, decision-making, motivation. These neurobiological networks are significantly changed with cannabis use and in cannabis use disorder. And when you look at cannabinoid receptors in the brain, this is with neuroimaging, as compared to a healthy individual on the right, we know that the cannabinoid receptors are changed over time. They even become reduced, and people even become tolerant with more and more cannabis use because there is cannabinoid receptor and the activity of these receptors change over time. So we know that, it's not just cannabis use disorder that's the issue with cannabis use, but perhaps other psychiatric disorders such as schizophrenia and other psychosis, depression, anxiety, and so on. We know absolutely that there's a high psychiatric comorbidity with a cannabis use disorder. Again, anxiety-related disorders, schizotypical types of disorders. But most of these disorders are diagnosed in adulthood. And we now know that most psychiatric disorders have a developmental origin. And that's one of the issues when we think about cannabis today and a lot of the debates about the legalization and the policies. We know that adolescent exposure to cannabis can have impact on adult behavior, but so can early life in utero exposure also have these long-term effects. So neurodevelopment is, I think, a really important aspect when we think about how do we legislate, how do we make policies that can impact on this. And the reason that it's so important in this aspect of neurodevelopment is that our endogenous cannabinoid system plays a really critical role in neurodevelopmental processes, from the making of the neurons and the cells as they migrate and form synapses into the networks, to even in late adolescence and early adulthood when the final maturation of our brain that takes place. And we know today that the cannabis is being used by pregnant people, also people who are breastfeeding, more people as cannabis has become legal. They're seeing that more kids are being exposed indoors to cannabis. And as I said, teens, that's when people experiment, and teenagers are definitely using cannabis. So again, the endocannabinoid system during neurodevelopment is a very dynamic process. It's not our endogenous cannabinoid system just stays there. During development, as I said, it's really critical for forming new cells as they're born and all throughout really that developmental process. An important aspect about the endocannabinoid system is that it's critical for hardwiring of the brain. So the cannabinoid receptors are expressed in a manner to help establish synaptic connectivity and the pathways in terms of in brain development. When we look at the human fetal brain that are exposed in utero, one of the things that we can see is that these genes and proteins that are important for cytoskeletal organization are indeed changed in those that were exposed in utero to cannabis. We see this also in the endogenous opioid system, our natural opioids that play a role in reward, response to stress and pain, and even correlates the amount of cannabis the mothers reported using. We even see it changes the dopaminergic system, that dopamine system that I showed you that before that's important for reward, goal-directed behavior, motivation, for many different processes, cognitive function. We can see that there are significant changes in the brains of these fetuses that are exposed to cannabis here in the striatum, this nucleus accumbens region, but also even in this region, the amygdala, that's important for emotional regulation. We see this significant impact that it's males that show the greater effect. But importantly, we can replicate these in our rodent models where we know the history of these individuals, we know the exact amount of THC that they consume. And so we know that THC can induce these effects, not just in human fetuses, but like I said, in causal in rodent models. But for us, an issue is, does it impact on human life, across the lifespan? Of course, that's difficult to follow people from in utero. My colleague Yoko Nomura, that we collaborate in these projects and looking at the effects of prenatal exposures across the life. And one of the things that we started to look at was how do we get an insights into what is happening in utero? And we started to look at the placenta, because we know that placental programming is essential for neurodevelopment, and that aberration of placental processing has been linked to psychiatric risk. And when we look at the placenta and study thousands of genes, we can see that genes, for example, the cannabinoid receptor, actually is changed, and it's reduced, correlated to the amount of cannabis the mother's reported using. But when you look at it in an unbiased manner, one of the things that we could see was that what was changed in a very dramatic manner was immune-related genes. No matter how we looked at it, genes that were increased or decreased related to some aspect of immune function. And we also saw that it was strongest in the males, and we could then also start to look at it in regard to the phenotype of the offspring of these individuals. We could see when these kids are about, for example, 4 years old, they have increased levels of the stress hormone cortisol. We could see that their clinical measures of anxiety, aggression were increased, associated with the prenatal cannabis exposure. We can also see that the immune-related changes in the placenta actually predicted the anxiety-related behaviors that we see in the offspring in the women who use cannabis predominantly. So those kinds of studies get a lot of attention in the media. Our studies like, cannabis use in pregnancy may lead to anxiety and aggression in kids and so on. But then you have consequences on women where they then arrest people for use of pregnancy during use of cannabis or during pregnancy, and even women who might not even have known that they were pregnant at the time that are being arrested. And for me, that's really not the take-home message, because one of the things that we also see is that excessive exposure to stress during pregnancy also impact on their children's clinical behaviors. In fact, the combination with prenatal cannabis had a synergistic effect. There's really, really strong effect on, for example, anxiety-related behaviors in their kids. So we're not going to arrest people during pregnancy because of excessive stress. So again, how are we arresting people for cannabis use? So in looking at children, we now see from that are exposed prenatally to cannabis, we can see early indication associated with that early life exposure. But there's so many confounds with studying humans. So what is there a causal relationship? And that's where animal models come in. We can study the placenta of animals that were exposed to THC, and we see very similar things in terms of the dysregulation in the placenta. Again, the immune changes predominantly in the males. And we can look at behavior, and we can see behavioral changes throughout their lives, not only on emotional dysregulation, depression like behavior, stress sensitivity to opioids later in life. But not all behaviors are changed. And that's something that's really important, because we're still trying to learn what are some of the issues by which cannabis have these long-term effects. But we can take the brains of these animals, we can sequence them and look at thousands of genes. And one of the things that comes up is that these protracted effects into adulthood relate in large part to these epigenetic changes. And epigenetics basically so we inherit our DNA sequence from our parents, the genetics, but the environment can change whether or not a gene should be turned off now is turned on, and change the function of that. And we can see that in adults, for example, with prenatal THC exposure, a lot of the epigenetic changes relates to aspects of, again, coming back to synaptic communication, synaptic plasticity. So our animal models and our human studies show us that prenatal cannabis, or THC, can have these protracted effects on behavior. They change the structure of neurons, the function of them, and these epigenetic dysregulation linked to synaptic plasticity. Adolescence is also, as I said earlier, an important window of neurodevelopmental vulnerability, because this is where there's dynamic processing going on in the fine-tuning of the final maturation of our prefrontal cortex. And the endocannabinoid system plays a critical role. We know, as I said earlier, this is a time period, adolescence, when many teens will experiment, not just with cannabis, but many different substances and other things as well. We see that there's an increase in cannabis use and vaping in particular during this time period in recent years. But, excuse me, there are a lot of debates about whether or not adolescent cannabis use really does anything. And one of the things that, for example, researchers in Europe for a long time have seen this association between cannabis use and psychosis. And, it's always talked about in terms of the more frequent the cannabis use or near daily use of cannabis, and also higher potency of THC. Here, Marta DeForti's team in England showed across Europe that potency is what really made a difference to showing increase in psychotic disorders in Europe. A recent study also verifies this in a manner in Scandinavia, in Denmark, where they have, their healthcare system is, socialized healthcare system, that they can look at cannabis coming into their society and the change in different disorders. And one of the things that they saw was that in recent years, that there's an increased association with cannabis use disorder and schizophrenia over the past 2 decades. Something is missing here, where it's mainly in the males, and particularly before the age of 26. After that, they didn't see an association with schizophrenia, and that about 30% of the schizophrenia cases were preventable by reducing cannabis use disorder. Also, there have been, old studies and debates again, what does cannabis do to the brain? Here, showing that early long-term cannabis use changes some of the structural organization of the brain, and especially with earlier onset of cannabis use. And recent studies in Europe, where they've done longitudinal studies looking at the same children over time, that they could see that there was altered cortical development in this prefrontal cortical region, and that this cortical thinning in particularly correlated with cannabis use and the teens' impulsivity. But there are a lot of debates even about those studies, because, the chicken and the egg. We know that early, as I said, neurodevelopmental things, not even cannabis exposure, I showed you stress. There are a lot of studies with early life trauma that changes the brain, that makes individuals more vulnerable. The environment, genetics, and the combination of the environment and genetics, these epigenetic changes, makes the vulnerability. And there are studies that do show, for example, the changes in the prefrontal cortex again can even predict initiation of cannabis use later in life. So it is very complex with humans. So once again, we can go to our animal models and look in the prefrontal cortex, look at these cortical cells, and these branches. These are where the synaptic connections occur between cells, and that are really critical for cognition. And we can see that in young adults that had adolescent THC exposure, the complexity of their cortical cells are reduced. And we can see that where they're reduced actually resembles just regular, stress. And we can go in, we can pick up these cells. Here, there's a laser, we pick them up, and we can then sequence them and look at the thousands of genes and see, is there anything that's relevant to, that can help us get insights into this long-term effects of adolescent THC exposure. And we can see that there's a complete reorganization of the gene expression landscape in these prefrontal cortical cells. In fact, only about 5% overlap to the normal developing prefrontal cortex during adolescence. Where they're changed in relation to morphology and these dendrite, these synaptic related cortical structural genes, but also to epigenetics. There's a profound significant change in regard to epigenetic landscape. And when you look at the relationship between these epigenetic related genes and these synaptic plasticity related genes in normal adults, there's not a great relationship per se, but in those that had adolescent THC exposure, there's a complete reorganization. And the gene networks that are changed actually are similar to those that we see in individuals who were diagnosed with schizophrenia. So we know that, from an epidemiological level, absolutely from individuals as well, that the aspect of high potency THC is really critical, that we see over time that there's this increase in THC content. And those are the issues that we have because the endocannabinoid system, like I said, it's really critical of regulating synaptic plasticity. But when these exogenous cannabinoids that are so potent that they're now changing the synaptic regulation to a pathological state. And those are the things that are associated most with psychotic symptoms, the anxiety and the cannabis use disorders. And adolescents and most adults don't necessarily titrate their use of these high potency products. I put this in here in terms of Jamaica, because I was born in Jamaica and people are like, oh, but people smoke cannabis all the time in Jamaica. They didn't smoke these cannabis in Jamaica. So when they looked at, for example, the cannabis products in the cannabis, I should say, products that's grown in Jamaica, in 2014, the concentration was 1.1%. It's now increased dramatically, even in Jamaica. So Jamaica was not a place that had this high THC concentration, and the ratio of THC to CBD, which was about 2-fold, is now nearly 200-fold. So the higher THC concentrations have also been shown to be associated, again, with these structural morphological changes in the brain. And that, again, this aspect of reducing the prefrontal, the cortex. So potency does matter. We can, again, this is Jackie Ferlin when she was a postdoc on my team. Now she's running her own lab. The question is, can we see it on again, even using animal models for causality, does dose matter? And we can see in animals that were given adolescent THC exposure, it's the high dose that they have protracted elevation of their stress hormone. And when they just have an acute isolation stress, they will self-administer for more rewarding. And we can look in the brain here, for example, in the amygdala, we can see that because amygdala is really critical for anxiety, emotional regulation, and even decision-making. And we can see that there are significant changes in their cannabinoid, their endogenous cannabinoid system. Cannabinoid receptors change, for example, and it correlates to their impulsivity. Human studies now can look at the microstructure of the amygdala, and they can see, yes, that indeed THC is related to changing the microstructural organization of the amygdala and the cortex, and they can see that it depends on dose. When we look at humans for their decision-making behavior, cannabis users, just like other individuals with substance use disorders, they're more risky. Our animal models with adults that had adolescent THC exposure, it's the high-dose THC that induces more risky decision-making. Low dose actually may even in one way, they stick to a strategy and can even have optimal under conditions. They also show with this high dose adolescent THC exposure, more impulsive behavior in adulthood. But we can also have the animals decide and consume their own cannabis, and so edibles. And we can see that animals, just like humans, they will separate into low consumers and high consumers. And the high consumers are the ones that show risky decision-making, just like the human cannabis users. But the thing that's interesting, a little stress, and they will consume more of those high-dose cannabis. So we can sequence again these amygdala in different brain areas, and we can see that high-dose THC and stress alters these cells, and many different cells. This particular one are called astrocytes, and they will change the structure of these cells. And these changes relate once again to decision-making and impulsivity. So high-dose THC is a major issue. But in most of the studies that we have in our human cannabis users, we're studying cannabis. But in our animal models, we can be very specific to THC in particular. But as I said from the beginning, we know the cannabis plant is complex, and it's not just THC, CBD, cannabidiol, which used to be much more prominent in the plant, that's now been decreased. That CBD might potentially have impact on the brain that may be different from that of THC. And in fact, we know that it's THC that really induces the reward, the intoxication, not CBD. And in terms of anxiety, we know that CBD may even reduce anxiety. THC increases anxiety, especially at high doses. And THC has a number of these side effects that we talked about earlier. When you look in the human brain, you can see that THC and CBD may even have different effects on the brain, opposite effects. For example, in the striatum of the brain related to aspects of even psychotics related symptoms, THC and CBD have opposite effects on brain activity. We can also see distinct effects in our animal models. So adults, for example, that were given THC or during adolescence, they will self-administer more heroin when they choose and can self-administer the drug. If we gave animals CBD, we were really surprised to see actually that it reduced their heroin-seeking behavior. And it reduced it even weeks later. So here we can say, that's one of the studies that people start to say, using our work, that CBD could perhaps, reduce opioid addiction. And we studied it and are still studying it in opiate users. Our first small study here looking at CBD in opiate users, double-blinded placebo control, and we looked at craving, and we showed them cues. And individuals who had received placebo and had seen, and when they saw the heroin cue, they craved, and CBD reduced that. Our animal models told us that it was having an effect even weeks after the last CBD administration. We brought people back in the lab a week later, it's still reducing their craving. Our human subjects told us there was anxiety that was really critical as well, because when they saw the heroin cue, their anxiety went up, CBD reduced that, and CBD even a week later was still reducing their anxiety. It's not just their self-report, we can look at their stress hormone level, and we can see when they saw the drug cue, their cortisol levels went up, and CBD reduced that. Similarly, when they saw the drug cue, their heart rate went up when they had placebo, and CBD reduced that. Now, again, we can use our animal models. Alex Chisholm, a postdoc in the lab, can replicate in both males and females that CBD reduces heroin seeking. And Jackie Furlan, we can see that in an anxiety model, that when they get a cue and had gotten the cue that associated with the anxiety promoting behavior, CBD will reduce that. And so now we're looking in the molecular mechanisms. We can look through the brain and we can see that under control conditions where genes can be increased and decreased, CBD will reverse it or normalize it, similar to the behavior that it's inducing. So these are some of the things that we see in regard to, the effects of cannabis on the brain, that potency and concentration matters, early exposure matters, frequency of use matters, even the biological sex may matter, psychiatric risk may matter, stress matters, the environment matters, and the cannabinoid matters. But it's also important that a lot of these things that matter are also what we see with other substances like alcohol, like tobacco. So it's really critical when we think about policy that these are things that we consider. So with that, my team, we look at preclinical and molecular effects in the human brain, and we also do clinical studies. And I'll stop there so we can discuss what the neuroscience may indeed have to offer in this discussion.
Thank you, Yasmin. That was fabulous and so interesting. I learn each time I hear you present. And we have a number of questions. One question was, how is the cannabis administered in these lab studies?
So cannabis was administered, in different ways. So different studies have had people, smoke a joint, or they can there are labs, I should say there are also studies that it's not even done in the lab. They are done because the federal laws that made it difficult for many people to get access to cannabis was also there for scientists as well. So it was illegal for studying, or it shouldn't, I mean, it's you can study it, but it's very challenging. So investigators have been very creative and even going out to the community where people may use cannabis outside and then come in to the research van. And so it's not that they're giving them necessarily always cannabis, but people are consuming it, and then they're looking at different aspects of toxicology and behavioral effects.
And what about the rodent studies?
Well, the rodents, they get it either through vaping. We even can inject it with them. Other people also use vaping and strategies as well.
Okay, great. And do you know of any studies looking at the brains of kids who've used cannabis medicinally for example, for epilepsy?
So cannabis for epilepsy, so that was one of the things that people used in terms of, changing laws, cannabis laws, when people saw that it was actually CBD, cannabidiol. It's not cannabis that alleviates epilepsy and especially these childhood, especially the rare childhood forms of epilepsy that for which the FDA has approved the use of CBD. So it's really important when we talk about cannabis, I think we should start talking about the cannabinoids, because THC is not a treatment for epilepsy. It's CBD. And so that's, to me, is very important. And that's why you do need more research to really understand. Many people think about the ratio of CBD to THC. We still don't understand that. And it's not that CBD will normalize every aspect of THC. It doesn't. In fact, under certain conditions, it might even exacerbate the effects of THC. So it's really important when people think about what's there for like epilepsy, that it's not cannabis, it's CBD.
That's really helpful. We have another comment. Someone said, "Fantastic talk, Dr. Hurd. What are your thoughts about using THC or THC that containing cannabis as a treatment for cannabis use disorder, somewhat similar to an opioid agonist for opioid use disorder?" What are your thoughts on that?
Yeah. Harm reduction to me is important. And I think that, I think we need to just take away the stigma about substance use and psychiatric disorders. If we really want to understand that this is a whole society problem, and harm reduction may be helpful for some individuals, but not all. So having replacement therapy with certain THC concentrations that we know are not as toxic to the brain as perhaps what someone might buy outside, especially these very high potent substances. I think that that's important for them to come in, get medical care, and get those if that's the treatment that they need. And we do hope over time that there will be non-addictive treatments, and that's why we've been trying to develop CBD as that. In addition to our studies with opioid use disorders, there are people studying CBD for cannabis use disorders, and some of that early data is very positive. And CBD is not intoxicating, and it can help give people their lives back.
And are you seeing the same genetic reorganization with CBD that you see with THC?
No. And, but so THC and CBD, they do act differently in the brain, even on the cannabinoid receptor. So THC is an agonist at the receptor. CBD actually does the opposite, maybe even a negative allosteric modulator at the cannabinoid receptor. And we see that there are different gene expression changes that go on with CBD and with THC. With THC, like I showed you, we see that it exacerbates like stress and early cannabis exposure, while in our models, CBD does the opposite and even reverses these the alterations that THC induces. So it's very different, but there's so much that we do not know. And that's why we need a lot of research, because people are using these substances. People are using concentrations that we had never, ever even thought people would ever use. And that's the issue and that's the problem.
Okay. And we have some questions getting into sort of the nitty-gritty of researching cannabis, which I think you're an expert in. There's one question about looking at studies and whether they take into account the impact of cannabis terpenes or the method of extraction for concentrates, whether it's an alcohol-based solvent versus solventless. I'm curious about, this person's curious about other variables besides THC and CBD that may be relevant.
Yeah, absolutely. NIH now has been calling for a lot of grant applications to study the other, the other 138 cannabinoids in the plant, and the other 300 or more other chemicals, including the terpenes in the plant. You need an army of researchers to understand the cannabis plant with all of the terpenes, with all of the cannabinoids. And because there now exists so many products, it is going to be impossible to look at the different combinations of all of these to really understand what is the impact of these substances. So if anything, that is the most scary thing, because when you think about alcohol, we understand ethanol, we understand concentrations. The cannabis plant is very different. Even when you think about cigarettes, the tobacco has many different chemicals as well. But really, when you think about nicotine, there are many more knowns, and people are not playing with the tobacco plants in the way in which they have done with the cannabis plant. So the different terpenes, the different chemicals, the over 100 cannabinoids, it is going to be very difficult to understand the impact, because in the natural plant, most of these are not in any high concentration, but now people are making them either synthetically or, even if they make plants that now make them more than they would be naturally. So that is the difference about the cannabis plant compared to alcohol and compared to tobacco.
Mm-hmm. Okay, thank you. That's really helpful. And we have some questions about the increase in THC. I think one of these I can answer. So there's one question about what is driving the increase in THC. And, I would just say that once most states legalized cannabis, there were no restrictions on THC level. So we had a bunch of new businesses flooding the market. And what happened is the most potent THC products got the most consumers. And so we've had more and more increases in THC, which has been really sort of toxic to the brain. There's another question about specific sources on the market where more natural, lower THC cannabis can be found. I would say that a lot of cannabis products indicate what the THC level is. Dr. Hurd, I'm curious, does that mean anything? I mean, there's so much variation in cannabis, and do you think that's a good indicator or something people should be looking at?
So at the end of the day, if we have a system where there's regulation, then you can trust what's on the label. And because there's regulation is what needs to be, just like, foods today, we know every single thing that's in the food product. The same thing should be there. And when they've tested a number of these products, even some of those companies, it doesn't have the same amount of THC that it says, or other chemicals, and they find other things as well. It's important to use legal stores to purchase things from, since they are regulated obviously more. But it is a challenge when the concentrations, and as you said, Stephanie, there was no limit put on what amount of THC should be there. So now, like I showed you, earlier in the slides, you have products that even can give up to 80–90% THC. Why? So that is the issue. And even if the product may be, you think you're buying 90%, but it really is 70%, that's still a huge amount. The original plants had 2 to 4% THC. And that becomes the challenge of how do you put the genie back in the bottle to regulate aspects of of concentration and dose and potency, when people now, want to sell the strongest and strongest thing, let's be serious. The stronger the more potent a product, the more concentrated, the more you're going to get clients, because you're going to increase their risk for becoming addicted to it. Plain and simple. So there's nothing different from any other drug, whether it's the cartel trying to sell more cocaine or anything, or or the opioids, where the synthetics now with fentanyl are much more potent. It's just to get more people, more clients.
Mm-hmm.
It's not about health. It's not about medicine. So if we think about medicine, and it's really important because the cannabinoids have medicinal properties that still need a lot of research to figure out dosing regimen, and which particular medical indication they will be good for. It will never be 50, 60, won't even 10% THC. It won't. That's not medicinal.
Yeah. So it sounds like THC has a lot of like neurotoxicity, but CBD, it seems like when you talk about it, it seems pretty safe. I mean, how safe is CBD? Can you use it in utero? Like if someone's pregnant, can they treat anxiety using CBD? Can they use it daily? Are there different types of CBD?
Every single thing you just said, we still don't know. So research on anxiety, there's more data definitely that CBD has anti-anxiety properties. What's the dosing regimen? We don't know. That's why we're doing research. The same thing for opiate use disorder. We have a big study that continued from our original study that we did, when everybody said, Yasmin, you're crazy. CBD cannot treat, addiction. And, we couldn't even get our papers published or anything like that. Now, people have replicated. And so we see, and we're trying to understand what doses may be relevant. Prenatal exposure or using during pregnancy, we have no clue. And again, dose does matter even for CBD. So even if CBD doesn't induce the intoxication, if it's a very high dose, people may feel like, the anti-anxiety effects and they think that's intoxication. It's not. But it's still important to understand that dose matters. So if on a scientific level and on the medical level, we don't know what doses to give to people yet, how can we say that CBD, even if it's, does not have the same toxicity as THC? I don't know if I'd be comfortable telling a pregnant person to use CBD because we don't know yet.
That's really interesting. And with other substances, do we see the same genetic reorganization that you talk about with cannabis, like say methamphetamines or fentanyl?
Yeah, all of these drugs, they change the genetic, the gene expression landscape. The question is, what are the biological systems that they're changing? How long do those changes last? And are those changes reversible? So one thing I will emphasize is that for me, I will say, studying the brain and addiction for decades now, what is clear to me is that these epigenetic changes give us an opportunity. Because unlike genetics, where you can't change your DNA sequence, epigenetic modifications are reversible. So I do think that it will be possible to, reduce the risk of a number of things that we see because they're epigenetically mediated.
Thank you. So I just want to say a big thank you to Dr. Hurd for an amazing presentation. I can't tell you how groundbreaking her research is and how lucky we are to have her here. And she's just a great human as well. So I want to say thank you to Dr. Hurd, thank you to Petrie-Flom and CLBB, and for the audience for joining us and for all of your great questions. I also want to thank my parents, who both are huge fans of Dr. Hurd, so they attend all of her lectures. Yes, I know. Great. So we will see you all again soon. I hope everyone has a good rest of the day. Thank you. Take care.
Thank you so much.